It's your call. I just wouldn't want to play.
You wouldn't want to mess with him.
Yeah. But I mean, it's gonna be hot, but
Yeah, it's my friends.
Oh. No.
I'll not come up with this.
Oh, gosh.
Oh my God.
We got stuck in the loop. Okay.
Okay, so yeah, this person may
Now?
need to move.
Okay, so we can move that chair somewhere else.
You can take the wheelie chair away.
Yeah, or just yeah.
Not wheelie chair.
Who's Daniel?
Daniel has the executive chair.
There we go.
Oh my God, I'm gonna see myself this whole
This
time.
person also not that good with computer.
I can minimize them if you
Okay.
want to watch.
Well, it's just kind of awkward.
It could, yeah.
Yeah, we can minimize it.
So how is it like seeing? Try to move it a little like closer. I'm starting to see the slide. What do you think?
Yeah.
Is that backwards?
Yeah, I don't know how
Oh, it's mirror.
projecting
Where are you shooting with?
webcam, YouTube webcam.
Hi, Andy.
Hey, how are you?
Good?
What? No.
I don't know how I am right now.
Hey, so I wanted to tell you, I saw Jean Moran on
Oh.
Friday. I
Yeah.
was chatting with her at lunch, and she says that degentrification is not an exclusively microbial process.
Oh,
There's
really?
actually plenty of evidence for.
Uh-oh.
So I think what you just say is it's often
Often.
or commonly,
Yeah. So okay.
and that's fine. But we should be clear, it's not exclusively. And in fact, she
So
was
I
saying that.
introduced it by saying microbes in the soil can remove nitrate via denitrification, and then maybe I should just. I do have a slide on top that says denitrification is a multi-step microbial mediated
Yeah,
process.
and you can just say usually.
Usually, okay.
That's fine.
Yeah.
Her point was just that they go to a lot of places where there's very small levels of denitrification,
Okay.
which they commonly attribute to abiotic processes. To
Okay.
something organic.
All right. So just one word, common.
It's commonly. I think it's fine.
Okay.
Just to be. I meant to send you an email over the weekend.
Oh yeah, no worries. That's fine.
Good.
Well,
Looks like
it's
you're all
fine.
set up here.
Oh yeah, so it's mirror. It looks fine on the.
It does. Okay. Yeah. Because it's like, and this is
Yeah.
mirror. The text is mirror image.
Oh.
But yeah, so
Huh.
But
But
when you're
only on
streaming
here.
it, when you're streaming it here, you can see it right. Okay.
And who's going to be?
Ryan, a few of my friends from
Oh, cool.
town. Chelsea, one of the Alex Steely and his
Nice.
wife. Delphine,
Oh,
Dustin.
oh my gosh.
Are going to tune in. It'll be 9
Oh my gosh.
p.m.
Yeah.
So keeping them up a little past their bedtime.
Yeah, they're just gonna have like a big bucket of popcorn. And just,
Well, they're probably at like
Pixel.
some pub.
yeah.
Probably they got it up on the
Got
screen.
them on the screen. Yeah. Yeah, London cares a lot about
So much more.
Yeah.
They don't really. The North is almost like-
Did you get it to turn this on?
Yeah, exactly. But it was really fun. No, I have like so much. How was your trip?
It was good to teach them all that stuff.
Yeah. I mean,
How was the?
So it's going even though, so you test, you minimize this, but it's so they can hear it back.
Yeah.
We're gonna cut this part out for the actual video. Unless you want it.
it is a very applicable issue. Yeah, exactly.
Yeah, for the lecture. Oh. Yeah.
She went to the first class. Her first.
Yeah.
That sounds fun. She starts.
Yeah.
So our girls were.
Yeah.
It was tough. My older daughter, she was like,
I drove back, it was like 3 hours.
that's right. It was like,
Took a nap.
but it's stressful.
Hopefully, the traffic was fine.
Then we're gonna sit like this for the entire conference.
Yeah. You were doing
Yeah, I did that cut through.
Yeah, I know.
But yeah.
I'm so tired. This is the worst thing. And then I didn't know the way back was bad too. Like it looked fine on 17, but I was like, is there another way back?
All right.
There wasn't so either.
Yeah.
Yeah. I didn't know.
All right. Welcome, everyone, to Sarah Beganskas' defense presentation. I'll let her get started in about 2 minutes, but I just want to do a quick introduction. I want to introduce Sarah's committee, Jason Gerdak from San Francisco State, right there, Noah Finnegan from our department. Thank you guys both for providing Sarah with so much support and encouragement and technical advice and all that. Sarah came here from Amherst College. She did 2 bachelor degrees, one in computer science and one in geology, and graduated magna cum laude. As an undergrad, she did some research in New Zealand, and then earthquake chased her away, and she came back to the US. Ended up doing some research as part of the Keck Consortium in Colorado. So she came in with some research background. And she's always been an ambitious student. So she already had 2 degrees, and then she got here and did additional coursework in physics and geophysics. She TA'd with Emily in 110C, which is quite a challenge, I think. It's a hard class, and in areas where she really didn't have much background before she got here. Also added expertise in geochemistry and microbiology, as you're going to see today. She's received a number of awards, and I'm just going to give you a partial listing. She's an NSF graduate fellow. She received the Wells Fargo Coastal Sustainability Fellowship this year, which is a competitive award. She received honors for her PhD qualifying proposal, 3 best student presentations at technical meetings, and there was a 4th one where I was standing there and they told us that they weren't going to give it to her again because she got it the year before, but she got the votes. Okay, so really she got the 4. They only gave her 3 because they stole one. And then she was a 2-time grad slam finalist here on that campus. So a lot of accolades. But in addition, she's been heavily involved, along with research and TAing with outreach and diversity efforts. She's one of the founders of GEODS, our geoscientists encouraging openness and diversity in the earth sciences, which is an award-winning campus program that has had I think, remarkably significant impact, considering it just got spun up so recently. It looks like it's going to continue for a while. She completed a 3-year professional development program through the Institute for Scientists and Engineer Educators, through the Center for Adaptive Optics. And she was selected to participate in 2 competitive programs in DC, one by AAAS and one by AGU for Science on the Hill, talking to staff of members of Congress, which is an important program for really getting science out in public view. She's also a judge repeatedly at science fairs, and then she worked helping to plan and coordinate activities with the Groundwater Association of California. So has really taken advantage of these engagement activities. It's a partial list, but it's quite impressive. She's got 1 paper published on one of the studies you'll see. There's another that's it's in review right now, and a third that's very close to being ready to submit. And she's collaborating with others on additional work. So she'll have other papers on which she's a co-author, and I'm looking forward to continuing the collaboration. Sarah has secured a postdoc at Temple University, working on an urban hydrology project that's really interesting, and that begins in August. I'm excited that she's going to stick around for a couple of months part-time and help us with the transition. And with that, I want to let her present her dissertation, Runoff, Infiltration, and Recharge Across Multiple Scales, Applications for Improving Groundwater Supply and Health. And I should have mentioned, she'll do her talk, we'll do public questions, and then the committee will get to questions. So, what do you got?
Well, thank you, Andy, and thank you so much, everyone, for being here today. I'm really excited to share some of my PhD research with you. And like Andy mentioned, my dissertation is focused on exploring the processes of runoff, infiltration, and recharge across multiple spatial scales. And my research also has applications for improving groundwater supply and quality. Groundwater is a critical resource upon which billions of people around the world depend for their fresh water. And this figure shows that global freshwater demand has more than doubled over the time frame from 1960 to 2000. At the same time, the rate of global groundwater depletion has also more than doubled over that time frame, from about 125 cubic kilometers per year in 1960 to over 280 cubic kilometers per year in 2000. And groundwater depletion occurs when the outputs from an aquifer, including pumping, exceed the inputs, primarily groundwater recharge. Here in California, groundwater supplies are also declining. This is a map of the state showing changes in groundwater level over a 10-year period from 2006 to 2016. And all of the red and orange dots, which you might notice are most of the dots, indicate significant decrease in groundwater levels over that time frame. Groundwater quality is also a growing concern in California. This figure shows average nitrate concentrations in groundwater over that same 10-year period. And in California and many parts of the world, nitrate is the most pervasive groundwater contaminant. It can have adverse impacts on human health and aquatic ecosystems. And for this presentation, I'll focus on nitrate as an indicator of groundwater quality. On this map, all of the red dots indicate concentrations above the drinking water standard. And over the last few years, from 2012 to 2015, a severe drought in California has intensified these issues and further stressed California's groundwater resources. These maps show the extent and severity of the drought for the last 3 years. One way to improve groundwater supply is managed aquifer recharge, or MAR. And MAR refers to a number of different strategies that route surface water underground into aquifers to improve water supply. For my dissertation, I focus on a specific type of MAR project, and that's distributed stormwater collection. Throughout the presentation today, I'll use distributed stormwater collection and DSC MAR interchangeably to refer to this type of project. And there are 2 important characteristics that define these projects. First, they collect stormwater runoff under the force of gravity. And so this schematic shows that when it rains, some fraction of runoff can be diverted into an infiltration basin. From there, the water can percolate into the soil and recharge the aquifer below. Another important aspect is that individual DSC MAR projects are typically very small, and the idea is to distribute many of them throughout a watershed, Collectively, many sites can have a large benefit on water supply, and it also ensures that benefit is distributed throughout a watershed. DSC-MAR projects also give us an opportunity to study many important hydrologic processes, including runoff generation, infiltration, and recharge. And they let us study these processes across multiple spatial scales, including individual field sites as well as entire watersheds. For my dissertation today, presentation today, I have two primary goals. I wanted to learn how distributed stormwater collection projects could effectively improve groundwater supply and groundwater quality. And I also wanted to use DSC-MAR projects as a lens to learn more about the processes of runoff, infiltration, and recharge. And in particular, I want to know what factors impact those processes across multiple spatial scales. Today, I'll share 3 different chapters addressing those 2 questions. First is a regional modeling study looking at the impact of changing climate and land use on regional hydrology. For my second chapter, I'll zoom in to a single DSC-MAR field site and look at runoff generation and infiltration dynamics during managed recharge. And finally, I'll present smaller scale field experiments that link hydrology, geochemistry, and microbiology during infiltration. And this is where the water quality aspect of my work will come in. So jumping right into chapter 1. Again, this is a regional computer model looking at the impact of shifting climate and land use on runoff generation and what that means for recharge. The basin in question is the Pajaro Valley Groundwater Basin, located about 30 minutes south of here, right along Monterey Bay. And in this region, about 95% of freshwater demand is met with groundwater, so it's a really crucial resource. And that's resulted in excessive groundwater pumping and declining water supplies. Having groundwater in this basin, though, is really important to support both the population of people who live there and a large agriculture industry. On this simplified land use map of the basin, the red areas indicate urban development, including the city of Watsonville, and the orange indicates agricultural development. Basins like the Pajaro Valley face many challenges on a regional scale, including changing climate and shifting land use, and both of those can have a big impact on regional hydrology. In terms of climate, that can affect the amount of rainfall as well as its intensity and spatial distribution. And it's not clear exactly what California's climate will look like moving forward, but many studies have suggested that the frequency of droughts, like the one from a few years ago, will increase moving forward. Human development, including urbanization and agriculture, can also significantly modify the landscape and affect regional hydrology. For example, impervious surfaces like buildings and roads and parking lots can change infiltration capacity across the landscape, and removing native vegetation and planting agriculture also adjusts the fluxes of evapotranspiration throughout a basin. And so on a regional scale, both changing climate and shifting land use impact how rainfall is divided between runoff, evapotranspiration, and recharge. They also affect the context and need, as well as opportunities for distributed stormwater collection. And for this project, I wanted to use a regional computer model to address 3 key questions. First, how do interannual climate variability and modern land use development impact runoff generation? And then what implications do those results have for groundwater recharge and distributed stormwater collection? I address those questions using Precipitation Runoff Modeling System, or PRMS. And this schematic shows all the processes that are included in this model. Climate data drives the model, and the water that falls as rain is ultimately divided into evapotranspiration, runoff, and recharge. To develop the model, I divided the Pajaro Valley into about 1,000 hydrologic response units, or HRUs, and these essentially act as model grid cells. I delineated them topographically to more closely match the pathways that water takes as it flows across the landscape, and so that's why they have irregular shapes. Then I characterized each of those HRUs with a number of different parameters, including soil and vegetation data. This is one example showing the percent sand in each HRU, with darker brown indicating sandier soil. For climate data to drive the model, I obtained 800-meter daily rainfall and temperature data for a 33 water year period. And this figure shows an example of one day of rainfall. When I overlay the HRUs on top, you can see the climate data is on a similar spatial distribution to the model. And finally, I calibrated the model using streamflow data. Unfortunately, there's no stream gauge at the outlet of the basin here, so I calibrated using streamflow data at this gauge, which incorporates a fraction of the basin. And this is a figure to summarize my calibration and validation results. On this figure, we're looking at total annual streamflow with the model data plotted against the observed data. The blue dots represent calibration and pink is validation. On the bottom is the monthly hydrograph, where the black line is observed data and the calibration and validation data are overlain. Overall, the model did a good job of representing streamflow in this part of the basin, and that's encouraging because streamflow integrates the signal of runoff from all the HRUs in the calibration area. So now I've given some quick background on how I set up the model, I'll dive into addressing these first two questions. How do climate and land use impact runoff generation, and what implications does that have for recharge? I'll start by looking at climate. To get a sense of how climate variability affects the regional hydrology, I created different climate scenarios using the historical data. And so each of these blue dots represents one water year from that historical record, and I chose years around the 20th, 50th, and 80th percentile to represent dry, normal, and wet conditions in this basin. These maps show mean annual precipitation for those three climate scenarios, with darker blue indicating more rainfall. And I also put the average rainfall for the basin in the upper right-hand corner. Now on the bottom, I'm showing model results for mean annual runoff generation for each HRU. On these maps, green and dark green indicates a lot of runoff generated, and the purplish pink means little to no runoff was generated. I also put some other information, the percent of rainfall that became runoff throughout the basin, and the average depth of runoff averaged throughout the basin in the upper right corner. A few key takeaways from the runoff data under these three climate scenarios. There's a greater fraction of rainfall that becomes runoff during the wet scenario compared to the dry scenario, 24% versus 7%. But during the dry scenario, there are parts of the basin that do have significant runoff generation, these green and dark green HRUs. It's just that significant runoff generation is focused within a much smaller area. And now I've added up here the total volume of runoff produced during each climate scenario throughout the entire basin. I found that collecting only a small fraction of all the runoff generated in the basin during the dry scenario can make a substantial impact on reducing groundwater depletion at about the 10% level. So overall, these results demonstrate that distributed stormwater collection can be effective even during dry times. At least in theory, there's enough runoff available to support these projects. However, especially during dry times, it's important to consider where the project is located because there's a lot of variability in how much runoff is available. And I'll return to that question a little bit later in this section. For now, I'll move on to looking at the impacts of land use. This is the more detailed land use map of the Pajaro Valley with urban development again in red and ag in orange. I wanted to get a sense of how urban and agricultural land use has impacted the regional hydrology. And so I created a pre-development land use scenario for comparison with contemporary land use. And to do that, I replaced all the urban and agricultural land use with a representative mix of native grasses and shrubs and forests. So here's the data you just saw for runoff under the three climate scenarios with contemporary land use. Now on the bottom is runoff for those three climate scenarios with pre-development land use. There's quite a difference both in the amount of runoff that's generated and also the spatial distribution and variability. Importantly, we note that about twice as much runoff is generated with contemporary land use compared to pre-development conditions, although it varies a bit by climate. And that suggests that if more runoff is being generated, less water is soaking into the soil and thus available for diffuse recharge throughout the basin. And so that suggests that there is likely less runoff, less recharge diffusely throughout the basin occurring under contemporary land use compared to the past. So far we've seen that climate and land use each impact how much runoff is generated in the basin and also the spatial distribution of that runoff. Next, I want to explore how simultaneously changing climate and land use can interact to affect runoff generation. There are many factors that impact how much runoff is generated in a certain location, and that includes the presence of impervious surfaces, soil properties, vegetation, and how much rainfall there is. So I want to know how climate and land use impact the relative importance of each of these factors for determining how much runoff is generated. And for now, let's focus on impervious surfaces. This is a map of impervious surfaces in the Pajaro Valley, with darker black indicating more impervious surface, and this is the city of Watsonville. I mentioned that human development can increase the amount of impervious area, which generally leads to more runoff generation. Because this is a land use parameter, I want to know how climate might impact the relative importance of impervious area for runoff generation relative to these and other factors. So this is a map of runoff generated during the dry scenario. Comparing the two maps, there's a lot of overlap between high impervious area and runoff generation. I use a Spearman rank correlation analysis to look at the relationships between different parameters and how much runoff was generated on average in each HRU. And this result shows that impervious area and runoff during the dry climate scenario have a relatively strong right correlation, a high correlation coefficient. On the other hand, this is a runoff map during the wet climate scenario. Comparing these two maps, you can see there's much less similarity between the two. And as we'd expect, there's a much weaker rank correlation between impervious area and runoff during the wet climate scenario. I forgot to mention that during the wet scenario, impervious area is less important, but other soil properties such as the storage and routing parameters were much more important for determining runoff generation during the wet climate scenario. And so these results demonstrate that impervious area has a larger impact on determining how much runoff is generated during a dry climate compared to a wet one. And ultimately that means that the climate plays a role in determining what the hydrologic response is to a change in land use that increases impervious area. Ultimately, that means that interactions between climate and land use are important to consider when thinking about regional hydrology and water management, because changing both at the same time can magnify the hydrologic response to either perturbation alone. And so now I want to do a quick recap addressing the answers to these first two questions. I motivated this chapter by talking about the impact that shifting land use and shifting climate can have on regional hydrology. My results demonstrate that considering interactions between these two factors is also important because they can magnify the hydrologic response. We also saw that under contemporary land use compared to pre-development land use, more runoff is generated in the basin and there's likely less diffuse recharge. This provides motivation for distributed stormwater collection projects that collect extra runoff to enhance recharge. And finally, we saw that significant runoff generation occurred even during the dry climate scenario. And that means that there are opportunities on a basin scale for distributed stormwater collection. However, during the dry climate scenario, that runoff generation was focused within a smaller area, and that highlighted the importance of choosing a strategic location for these sorts of projects. And that's exactly the question I want to address for the last part of this chapter. Where are the best locations to collect runoff for MAR during the different climate scenarios? So there are many different ingredients to a successful DSC MAR project. First, we want to consider soil and bedrock properties. So how quickly can water pass through the soil, and does the location overlie an aquifer? There's also properties of the aquifer itself. So how much storage is available, how quickly can water spread out once it reaches the aquifer? And finally, the supply of water for managed aquifer recharge, which in this case, I'm focusing on hill slope runoff. We have spatial maps that represent all of these properties, and by overlaying them, we can try and find locations that have relatively favorable values for all of them, as shown in the schematic here. This is a map of the Pajaro Valley showing in light blue areas that are suitable in terms of soil, bedrock, and aquifer properties, and also generate at least 13 centimeters a year of runoff during the wet climate scenario. And I chose that cutoff because it's a significant amount of runoff that's enough to support a DSC MAR system. We can narrow that down further. These are areas that meet those criteria during the normal scenario. And finally, in the darkest blue, those areas meet the criteria during the dry scenario. Overall, this map demonstrates that there are many opportunities for stormwater collection around the Pajaro Valley during all three climate scenarios. And in addition to these more obvious larger swaths of dark blue, there are smaller groups of pixels all around the Pajaro Valley that are suitable during the normal and dry scenarios. And that's important because it really only takes a small amount of land to build a successful stormwater collection project. To illustrate that, we can zoom in on this red square to look at the first DSC-MAR field site in this basin. This red shape is the infiltration basin, and this project, when successful, can address groundwater depletion in this basin on the order of about 1%, which is impressive considering the small size of this system. That means building 10 or so similar projects around the Pajaro Valley could have a really substantial impact on improving groundwater resources while taking up a very small area. It also means that the areas I've delineated here could be further screened for other considerations like land ownership, ecological factors, and other relevant considerations. So ultimately, our model results show that there are basin-wide opportunities for distributed stormwater collection to improve groundwater supply around the Pajaro Valley during the 3 climate scenarios I tested. And so how well do these model results translate into the real world? That's the question I'm going to address for chapter 2, where I'll zoom in and focus on field data from the site I just showed in the previous slide. At this field site, there are 2 primary questions I want to address. First, I want to put those regional modeling results in a more local context and see what kind of water supply benefit can be achieved using hill slope runoff in practice. And in particular, I want to see how well this works during a dry period, the drought I mentioned in my introduction. And second, I want to know what happens to that water after we collect it. So what factors impact infiltration dynamics at this field site? This is an aerial photograph of the field site showing that hill slope runoff from 172 acres of farmland is diverted into a 4-acre infiltration basin. And otherwise, this runoff would be diverted into a storm drain, into the Pajaro River, and out to the ocean. The goal for this field site is to collect and infiltrate 100 acre-feet of runoff each year. And as I mentioned in the last chapter, that can address regional groundwater depletion by about 1%. I've worked at this field site for the past 6 years to measure precipitation, runoff generation, sediment transport, and infiltration rates. So now that you know a little bit about the field site, let's jump in and address this first question: what water supply benefit can be achieved in practice through distributed stormwater collection? To answer that question, I want to know how much runoff is collected each year. And to do that, I can measure the flow rate of water through this culvert that delivers water into the infiltration basin. To do that, I install pressure gauges that tell us the depth of water in the culvert. From there, I can calculate the wetted area and wetted parameter of the flow, and then plug into the Manning equation to calculate the volume flow rate. And then I can turn that into a total volume of water that's flowed through the culvert over the course of the year. This figure summarizes 6 years of data from the field site, showing how much runoff was collected each year and how much rainfall there was. This horizontal line represents the project goal, 100 acre-feet, and this vertical line is the mean annual rainfall. The 4 years in red here represent the regional drought that I mentioned in the introduction. And this project exceeded its goal during the 2 non-drought years and during one of the 4 drought years. So it seems to be successful in practice. This year in particular is an exciting result because I'll remind you that this is this extent and severity of the drought in 2015 in California was very intense. And so at a time when California's groundwater supplies are very stressed, it's encouraging that distributed stormwater collection can still collect a large volume of runoff to improve groundwater supply. Ultimately, the success of this project provides validation for our regional models, which found that DSC-MAR could be successful in this basin under a range of climate conditions. But what's so special about 2015? Why was so much runoff generated that year compared to the other 3 years of the drought? Well, most of the rainfall and most of the runoff in that water year were generated during a single series of storms that lasted for about 10 days. 3/4 of all the runoff collected during that year were collected in about 7 days, and about 1/4 of all the runoff for the year was collected in less than 2 days. We can compare that to 2013, a year which had a similar amount of total rainfall, but about 1/4 of the total runoff compared to 2015. In 2013, there was a larger number of storms that were each less intense and shorter in duration than the storms in 2015, and clearly that had a big impact on how much runoff was generated. These results demonstrate that total annual rainfall is not necessarily a great predictor of how much runoff will be collected, because rainfall intensity and storm duration are also important factors. And that means that having a dry year like a drought doesn't preclude there being opportunities to collect runoff during intense rainstorms. So both my regional modeling and field results demonstrate that stormwater collection can significantly improve groundwater supplies even during a drought. And that's because intense rainfall can occur whether or not a year is wet or dry. Moving on, let's explore what happens to the water, the runoff, after we collect it. So what factors impact infiltration dynamics at this field site? A large control on infiltration rates during managed aquifer recharge is often the accumulation of fine-grained sediment. So you may have noticed from the photos I showed earlier from the field site, the runoff that we're collecting or that's being collected is a really nice brown color. And that's because it's carrying with it a lot of sediment. So that sediment can accumulate, and it's been documented at other managed aquifer recharge field sites to clog up the soils, reduce infiltration capacity, and ultimately limit the effectiveness of these systems in the long term. And so I wanted to evaluate what impact the sediment might be having at this field site. To do that, I deposited or deployed these trays at the start of each wet season, and then collected them in the spring when they look more like this. And my calculations show that a lot of sediment is accumulating each year, up to 420,000 kilograms per year of sediment, which works out to an average sediment load of up to 11 grams per liter. And that's actually not so high for an agricultural region like this. The loose, well-drained soils that work well for growing crops also tend to be transported very easily. But understanding the impact that the sediment has on infiltration also requires knowing something about its texture relative to the infiltration basin subsurface. And so I'm going to show grain size data for soil samples collected first from the infiltration basin subsurface. We collected these soil cores using a hand auger between 1/2 and 2 meters deep below the surface, and then samples from these accumulating sediment. So these are plots showing grain size distribution. On the x-axis is grain size on a logarithmic scale, with finer grains on this side and coarser grains on this side. The y-axis is volume fraction as a percent, so it's the percent of grains that fit into each of those different grain sizes. Each individual gray curve represents the distribution of grain sizes for one sample, and these darker, thicker curves represent the average for the 3 years for which I collected this data. Comparing these distributions to these distributions, there is a much higher fraction of finer grains and a lower fraction of coarser grains in accumulating material compared to the subsurface here. And we also see evidence for fine grains accumulating in the subsurface over time. So over time, the fine fraction increases and the coarse fraction decreases. This ultimately means it's not looking good. There's a lot of sediment accumulating, it's relatively fine grained, and it's accumulating both on the surface and in the subsurface. But let's see what impact this might actually be having on infiltration rates. This figure shows 5 years of data. The green data on the left axis is showing cumulative runoff collected. So each of these vertical lines represents a time when a storm created runoff and it flowed into the basin. On the right-hand axis in purple is average infiltration rate, which I calculated using a mass balance for the basin. There's a lot of variability from year to year here, but ultimately during big storms, it's not uncommon to have infiltration rates between 1 and 2 meters a day, and often much higher than that. So there really doesn't seem to be a long-term decline in infiltration rates at this field site, despite that accumulation of sediment, or at least not yet. Rather, we see that the fastest infiltration rates tend to occur during these largest storms. And so that suggests that at least the way this field site is currently operating, the intensity of rainstorms is having a bigger impact on how quickly water infiltrates than the texture of the soil, at least for now. We can dig a little deeper into this question, though, by measuring infiltration rates at specific points, not just averaged across the basin. And we do that using temperature data. So there's a photo of me installing some thermal sensors below the surface, and here's a schematic of what our instrument looks like. So we installed these 2 thermal sensors at a known depth distance below the surface. And then as water infiltrates through the soil, it carries with it heat from the surface. We expect that those temperature records will have a daily fluctuation to them that mirrors the night-day cycle at the surface. And because the sensor is shallower, we expect that it will have a higher peak and that the peak will arrive a little bit sooner than the deeper sensor. So by quantifying that phase shift and amplitude reduction, we can calculate how quickly the water was infiltrating and carrying heat vertically between those 2 points. These are maps of the infiltration basin showing 12 different points with the infiltration rates that we calculated. Green and blue indicate faster infiltration, and yellow and brown indicate slower infiltration. And you can see just by looking at these maps, there's a lot of variability both in space and in time in terms of infiltration rates. We only have 12 points here, and it's clear that these 12 points don't fully sample all of the variability that might be occurring within the basin. Now pick a few locations and compare how the infiltration rate changed on December 14th compared to a few days later. There's often really big shifts at the same location, up to an order of magnitude over just a few days. And that indicates that there are likely many factors at play controlling how these infiltration rates are varying. But it means that the long-term accumulation of fine-grained sediment might not be dominating at least these shorter scale fluctuations. And so fully understanding these dynamics is definitely an area for future work. We also noted that these point-specific rates tended to be slower on average than the basin average rates that I calculated. And that suggests that there are faster infiltration pathways dominating the infiltration flux that these 12 points are not sampling. So here's a photo of the infiltration basin at the end of a wet season. There's this vegetation that grows anew each year, covering the entire base of it. And we think that the annual growth of this vegetation with roots that penetrate to the soil and perhaps some help from some burrowing animals who live in the area could help develop and maintain faster infiltration pathways. So those processes create ways for water to quickly get through that shallow subsurface and mitigates possibly the impact of that fine-grained sediment accumulation. So our results show that infiltration rates vary both in space and time at this field site, and there are many potential factors that can play a role in determining those variations. We'll see in the next chapter that those fluctuations in infiltration rate are really important for water quality improvement. So for my final chapter, I'll present some smaller scale field experiments that seek to better understand controls on nitrate removal during infiltration. So way back at the beginning of the talk, I introduced that nitrate contamination in groundwater is a significant challenge in California. When water infiltrates into the soil, microbes that live in the soil can remove nitrate from the infiltrating water via a process called denitrification. And this is data from an active MAR field site in the Pajaro Valley showing evidence of denitrification. This data showing nitrate removal rate plotted against infiltration rate. A few important takeaways from this data. At least in this study, no nitrate removal was observed and infiltration rates above about 0.9 meters per day. We think this is because microbial denitrification requires that there's little to no oxygen present. And at some point, if water is infiltrating too quickly, the delivery of oxygen will be so fast that anoxic or suboxic conditions never develop. We also noted that about half the time that infiltration rates were below this threshold, nitrate removal still didn't occur. And this inconsistent behavior suggests that other factors in addition to infiltration rate help determine how much nitrate removal will occur. To put this data in context, MAR projects are designed to infiltrate water quickly. A typical range is between 0.5 and 2.5 meters a day. Most of that range is above this 0.9 meter a day threshold. And so a key question that I want to address is, how might nitrate removal be enhanced during rapid infiltration like that you'd see for an MAR project? In addition to requiring little to no oxygen, microbial denitrification also requires that there be abundant carbon. And so one approach to removing nitrate from shallow groundwater has been to install a vertical carbon-rich amendment in the aquifer. And that can be made of any carbon-rich material like wood chips, biochar, and so on. As groundwater flows through that material, there are favorable conditions for denitrification here, and water quality is improved downgrading it. So we wanted to know, would that approach work during managed aquifer recharge? If we installed a horizontal amendment made of carbon-rich material, could that result in enhanced nitrate removal? To test that idea, we designed small field-scale experiments to link hydrology, geochemistry, and microbiology in the subsurface during infiltration. And there are 3 questions I wanted to address. First, does the carbon amendment result in enhanced nitrate removal? And then how do the wood chips affect the patterns of nitrate removal as well as microbial ecology in shallow soils? This is a cross-section representing our experimental design. So we dug 4 meter square plots in the field and filled them with water so that water would continuously infiltrate, representing conditions you might see during managed aquifer recharge. Of our 4 plots, 2 of them look like this, which had native soil right below the plot. And another 2 plots had a layer of wood chips like shown here. And now I've added on some of the instrumentation we used for these experiments. These are piezometers, which we use to sample fluid from the subsurface as it's infiltrating. And these are the thermal probes I mentioned in the last chapter, which are used to calculate infiltration rate. Here's a time-lapse video of one of our plots in action. This is sped up, so it's showing 1 frame every minute. And to operate the experiment, we pumped groundwater on site, which had very high nitrate. And then the float switch helped keep the water within a finite range to continuously infiltrate water for weeks at a time. A few more photos from these experiments. We installed the 4 plots relatively close to each other. And so in this photo, you can see 2 different plots. Here's a photo of me and Galen adding wood chips to one of the plots. And we also went out each day during the experiment to collect fluid samples from the surface and subsurface. The soils at this field site were very sandy, and that resulted in very rapid infiltration. This data is showing infiltration rate over time for an experiment with native soil in blue and wood chips in red. This horizontal dashed line is that 0.9 meter a day threshold from the beginning of this section. And so these experiments present a good way or good setting to look at nitrate removal during very rapid infiltration. All right, so now that we know a bit about the experimental design, I'll start by addressing this first question: are the wood chips associated with enhanced nitrate removal? To address that question, I calculated the nitrate load both in the surface water that's inflowing and then also in this deepest sample. The nitrate load is a function of both the concentration of nitrate in the water and the rate at which it's flowing into the ground. And a difference between these two can indicate a net removal of nitrate. So these figures are showing the nitrate load over time for inflowing water. Again, our native soil on top and wood chips below. The concentration of nitrate in the inflowing water was relatively constant, but the infiltration rates varied over time, and that meant the nitrate load also varied over time. Now, the solid blue line represents the nitrate load in that deepest sample, and I've highlighted the areas, the difference between in gray to represent addition and light blue to represent removal. Overall, there wasn't much consistent nitrate removal during these experiments, and for this one, it added up to about 1 gram of net removal over the 2 weeks. However, below the wood chips, there was consistent nitrate removal after a few days spin-up period. So here's the removal the net nitrate load in the deepest sample, and I shaded in the removal in light red. Overall, it added up to about 20 times as much nitrate removal compared to the native soil. And although 20 grams might not seem like a huge amount, the removal rates we observe actually scale up to a sizable amount. This works out to about 15 kilograms of nitrogen removed per hectare of infiltration per day, which is 5,500 kilograms per year. And to put that number in context, here in California, a hectare of strawberries will typically receive 220 kilograms of nitrogen fertilizer each year. And so we do see that nitrate removal was enhanced after passing through the wood chip layer. My next 2 questions will explore a little bit more about how and why that's occurring. So first we'll look at some geochemistry data to look at the patterns of nitrate removal in the soil below wood chips and in native soil. So we're going to look at concentrations of nitrate and carbon in all of these different samples from different depths. And I want to note that we're only able to collect samples from these piezometers when the soil around them is saturated, and that affects the maximum depth to which we can sample in these experiments. So here's some data from the native soil experiments. This is showing data for the surface samples and then 2 samples from the subsurface. These gray bars coming out to the left is the concentration of dissolved organic carbon, and the teal bars going out to the right are concentrations of nitrate. Ultimately, there's not much change in dissolved organic carbon with depth, and there's only a very small amount of nitrate removal on average. Again, things look different below the wood chips. So this is data for the surface sample, a sample within the wood chip layer, and then we're able to collect 3 samples from the subsurface. DOC tends to increase with depth below the wood chips, and we also see more significant nitrate removal compared to the native soil. I don't have time to show it today, but isotopic data is consistent with denitrification being the mechanism for nitrate removal here below the wood chips. To get a more general sense of how the wood chips might affect nitrogen cycling dynamics, we can put data from this study into a wider context. So here's the figure I started this chapter with. and I've added our data in for comparison. So for native soil, we always had infiltration rates above this threshold of 0.9 meters a day, and there was little to no nitrate removal. So that data is consistent with the paradigm I presented at the beginning of this section. Below wood chips, though, we see some different behavior. There still appears to be a threshold, and it appears to have increased to about 2 meters a day. So above that threshold, there's still little to no nitrate removal occurring. And that suggests that although the wood chips may enhance nitrate removal, there's still a limit on how quickly water can be infiltrating. On every day with infiltration rates below that 2 meter threshold, nitrate removal occurred. And that's more consistent behavior than was observed in native soil. Clearly, there's more work to be done to further characterize this space, but our data does demonstrate that the wood chips appear to fundamentally shift how nitrogen cycling works in the subsurface. And so they're associated with more consistent nitrate removal that occurs over a wider range of infiltration rates. So to summarize the geochemistry data, we saw that wood chips provide carbon to the subsurface and are associated with more consistent nitrate removal. Finally, I want to know how wood chips might be impacting microbial ecology in these soils. And that's because denitrification is a multi-step, often microbially mediated process. It's the progressive reduction of nitrate all the way to nitrogen gas. And there are many groups of microbes that are capable of carrying out individual steps within that denitrification pathway. So I wanted to know whether microbes with those capabilities were present during this experiment, and if so, how might the wood chips impact them? To do that, I collected soil cores before and after infiltration during these experiments. And we performed 16S and 18S rRNA gene sequencing on some of those samples. This data analysis could tell us the relative abundance of microbes within each sample. And I divided the data into 4 statistically similar groups. So we have native soil before and after infiltration, and the wood chips before and after infiltration. Here's some of the microbiology data. Along the bottom here is the different groups of microbes, but don't worry too much about their specific names. The top row here in red is for wood chips, and the second row in blue is native soil. And these are both for samples collected before infiltration. The size of each circle represents the relative abundance of that group of microbes in those samples, and a larger circle means higher abundance. These little arrows mean that there is a small but non-zero relative abundance for that group, and then the white space indicates that there is none detected. Comparing these two data sets, there's really little differences between the microbial communities before infiltration for both experiments. Now I've added data for after infiltration, again with the wood chips in red and native soil in blue. For native soil, there doesn't appear to be a large shift in relative abundance after infiltration. But below the wood chips, there is a large shift, and in particular, these groups towards the right-hand side are enhanced relative to the relative abundance before infiltration. Many of those that are enhanced have the capability of carrying out different steps in the denitrification pathway. And so now I'm highlighting all those that are capable of carrying out nitrate reduction, the first step. These are the microbes that are capable of carrying out the second step, the third step, and the fourth step. And so overall, most of the microbes that were enhanced below wood chips contain the capability to carry out at least one of these 4 steps in denitrification. We also see that most of them were present in the soil before infiltration and were not enhanced in native soil. That tells us that wood chips may be contributing to favorable conditions to denitrifying bacteria in the soil below that wood chip layer. And it's ultimately consistent with the geochemistry data indicating that denitrification was enhanced below wood chips. And so to summarize again, the wood chips may be contributing to favorable conditions for denitrifying microbes. And this finding provides some sort of explanation for the results I presented earlier in this chapter. So for the last few minutes, I want to tie together my results across multiple scales and review some of the main conclusions from my dissertation. In my first chapter, I presented some regional computer models looking at the impact of climate and land use on the regional hydrology and in particular runoff generation. I showed that there's more runoff and likely less diffuse recharge today compared to the past as a result of shifts in land use. I also demonstrated that the impact of simultaneously changing climate and land use can magnify the impact of those regional perturbations and how important it is to consider the interactions between the two. Both the regional models and field data showed that collecting runoff to enhance recharge can be an effective strategy even during a regional drought. And the field observations also showed that infiltration rates varied very much in space and time, and identified several factors that could contribute to those variations. In the third chapter, I showed that variations in infiltration rate are really important for water quality because infiltration rate was a significant control on how much nitrate removal occurred in the subsurface during infiltration. And my field experiments from chapter 3 ultimately provided direct links between hydrology, geochemistry, and microbiology in the surface during infiltration. Overall, I conclude that managed aquifer recharge projects that collect stormwater runoff and use a carbon-rich soil amendment can be an effective way to improve both groundwater supply and groundwater quality. And that's really important in a state like California, whose groundwater resources are so stressed. It's also more broadly applicable around the world. Recall, we have increasing freshwater demand and an increasing rate of global groundwater depletion. On a broader level, my dissertation also demonstrates the power of combining interdisciplinary methods across multiple spatial scales. And doing work like this has potential to learn about fundamental research questions, including the hydrologic processes I identified here, and also to develop effective water management solutions. Whew. Okay, so before I finish, I want to spend a few minutes acknowledging a lot of people. And I'll start with my funding sources to support me and my research. That includes the NSF, Gordon and Betty Moore Foundation, UC Water, California Coastal Conservancy, EPA, and the Recharge Initiative. So I'm really grateful to have had a lot of funding to support this work. And then I have a lot of people to thank. I want to start by thanking Andy, my advisor. Thank you for giving me the opportunity to come here. Being an amazing mentor for the past 6 years, you're really very patient and thoughtful and really a great leader, very inspirational, both within our lab group and in the water community. And so I really appreciate the support you've given me going above and beyond, I would say. I also want to give a big shout out to my fellow Hydro Group members who have really been my collaborators and my friends over the last 6 years. Especially a big shout out to Galen, Tess, Walker, Kyle, and Ryan, who in addition to being co-authors on this work have put in countless hours with me with both technical and personal issues. And so I'm really grateful to have worked with such a great lab group for the past 6 years. I also want to acknowledge my dissertation committee, Noah and Jason. Thank you for all your technical input and support over the years. I feel lucky to have you on my committee, and I'm grateful for you taking the time. I'm also lucky to work with a number of diverse collaborators outside the Hydro Lab Group, and I'll call out a few of them today. First, there's the Saltykov Lab Group, including Katie and Nicole. Thank you guys for helping me with the microbiology stuff. I really learned a lot from you. And then outside of UCSC, I did collaborate with the Resource Conservation District of Santa Cruz County, especially Sasha Lozano, and the Pajaro Valley Water Management Agency, especially Brian Lockwood and Casey Musil. And having input from these folks, having their perspective has been really helpful for me and I think really improved my work. So I'm grateful to have worked with them on the applied side of things. I also want to give a big shout out to everyone who's been involved in the GEODS community. So everyone who's been a leader in the past and present. GEODS has been a really meaningful part of my grad school experience, and I'm really proud of everything we've achieved, and I'm excited to see where it goes moving forward. So really heartfelt thanks to everyone who I've worked with on that. It's been really, really great. We're also really lucky in the EPS department to have wonderful technical staff and administrative staff. So thanks especially to Rob Franks in the Marine Analytical Lab and Ed Boring on IT. And I especially want to call out Jennifer Fish and Amy Kornberg from the front office. They've really helped me out with so many logistical things over the years, and they really do care about the grad students. We're lucky to have them. I can't call everyone out today, but I'm also lucky to have a good support network of friends in Santa Cruz and beyond. So thanks to everyone for support and for fun times over the years. And I also want to thank my family. So Mom, Dad, Michelle, Tommy, thank you so much for being here today, coming all the way from New York. Your support and encouragement really gets me where I am today. So I'm really happy you could be here, and I'm looking forward to celebrating with you. And then last but not least, I want to thank my amazing fiance, James, for really being my partner throughout all this and helping me along every step of the way. So thank you, everyone. Thank you, everyone, for coming today. And I'd be happy to take some questions now. Dina. Hi.
I noticed that you did your experiments with the wood chips and then with the soil, and how would that impact selecting location?
So my results don't directly suggest what would happen at slower infiltration rates, but that's something that other members of my lab group have been working on. And so we've done additional field studies and column experiments at a range of soil textures and a range of flow rates. And we've had some consistent results, at least in the geochemistry, showing that the wood chips are associated with enhanced nitrate removal compared to the native soil. And so my pilot study kind of demonstrated that, and there's definitely further room to kind of fine tune and to see how far those results are consistent across other soil types. Yeah. Slavic.
So is the bottom of the MARs being managed in any way to increase infiltration, or is it just overgrown and left untouched?
At the field site that I've been, yeah. So each year they actually till the whole thing, and so the vegetation dies over the summer. It's too dry. Typically, they till it all under, and so at the start of the wet season, it's just totally tilled soil, and then they don't actively manage it to increase infiltration rates. It happens to be that the soil there is relatively sandy, and so it does have favorable infiltration capacity, but it's not actively managed, at least not yet.
Sarah doesn't know this, but
Oh.
the grower called us and asked us to get our equipment out of there so they can scrape it.
Oh, they're going to scrape it this
We're
year.
gonna scrape it the
Wow.
first time, I know.
Wow.
So that's a big,
Maybe our data.
Yeah. so that's good.
Gophers too.
The gophers,
Yeah.
yeah.
So,
Dan.
Are all the microbes that you found presence of, were those present on the wood chips like out of the bag, or did they have to like inoculate the layer chips?
No, so we think that the fact that they were present in very low relative abundance before infiltration suggests that the wood chips merely created favorable conditions for them to grow. We're doing some work now to try and actually sequence the wood chips themselves to see if they might be contributing any microbes. What else did I want to say about
And all your samples came from the soil.
Yeah, all the samples are from the soil.
Okay.
Yeah, absolutely. What was there another part of your question? I thought there was something else I wanted to say. Okay. Jim, yeah.
Yeah, so I guess I'm trying to get a sense of scaling up in that.
Mhm.
There's some percentage of runoff you calculate for dry years versus wet years. And for this to be an effective system at sort of recharging aquifers, do you know, can this be scaled up large enough to, what scale does it have to go to? Or are we already there with these recharge ponds?
Yeah, so I think ultimately this type of approach is just one part of a broader solution to improving groundwater resources. And the Pajaro Valley Water Management Agency is working on a multifaceted approach, including other larger infrastructure projects and conserving water, reducing groundwater demand, reducing water demand. The idea for distributed stormwater collection projects is to have many small sites distributed throughout a watershed. And so individually, each one can be located within a single property, not take up much space. And if they're put in a strategic location that will generate a lot of runoff, collectively that could have a really big impact on the water supply. And so I think you can think about scaling it up both in terms of the size of the system, but also just the number of projects that you build.
It's a related question. So
Okay.
it seems to me that most of the runoff happens like in cities, in built areas, and that's not a solution for them. And you already said that there's multiple solutions and whatnot. If you had a limited budget and you had to recommend something, since you're going to DC to
going to the city
about policy stuff, how would you kind of balance these effects, collecting stormwater in the city rather and, cleaning, recycling, using, or even diverting it to a managed aquifer?
Yeah, so within cities, actually, there's even smaller scale managed aquifer recharge projects, on a, meter or so scale that can actually infiltrate significant volumes of water when land area is scarce. And it's funny, you mentioned my future work. The projects I'm going to be working on are a similar idea that they're seeking to collect runoff, but the idea is to mitigate flooding, not to increase groundwater recharge. And so I think the same approach can address multiple issues at once, and that kind of determines what factors you prioritize in choosing a location and a specific approach. And so I mentioned my work has focused on infiltration basins, but there are other ways of getting water from the surface to the subsurface if you don't want a large body of standing water. For example, dry wells, infiltration trenches, that sort of thing. And so
Even infiltration in like paved.
permeable pavement, yeah, absolutely, yep. And so all of those things can,
Ditches and
Yeah.
sides of roads.
absolutely. And so that's one of the benefits of collecting this runoff before it reaches a stream rather than diverting it from a stream, is that you're collecting it during these large flood events and mitigating impacts on the channel during a flood stage. And so I think at all scales, from permeable pavement up to infiltration basins, reducing the amount of runoff during a flood that reaches the channels is going to be a good thing, both for the channel, for reducing floods, and for improving groundwater supplies. So, yeah, Daniel.
You said that the sediment load accumulated there, and so I'm worrying back to the scaling question. Did
Yeah.
a smaller basin receiving less runoff receive proportionally less sediment, do you think? And could they be better to maintain or manage, not have to come in straight from?
So, I guess in our study we saw there was a big range in the sediment load of the runoff, and so I'm not sure a smaller system would have less runoff, but it would also, if it had a similar sediment load and a similar proportional area of infiltration, you might see a similar depth of accumulation each year. And so I think mitigating that is really a challenge and needs to be considered individually at each field site. We've also done some work looking at the drainage area in addition to the infiltration basin. So you want an infiltration basin that has relatively coarse soil so you can infiltrate the water quickly. But the properties of the drainage area also matter, because that's the material you're going to be eroding and transporting into the basin. And so that's something you can also kind of get at with these regional analyses too. So there's certainly a lot to consider. And there are strategies to mitigate that sediment if it is accumulating. So yeah, good question.
All right, well, let's thank Sarah once again. May's gonna ask her questions, and within a couple of hours, she'll be out. If you have more questions, just hang out.
Thanks everyone for coming. Yeah.